Hi everyone, welcome to this week novo protein engineering seminars. This week we have Laurel and Thomas, both from Institute. They're going to present their paper entitled Flexible Antibody Design with Multimodal Bayesian Flow Networks. You can take it from here.
Awesome. Well, I will take it from here to start. So thanks very much for joining everyone. As you go, I'm Tom, so staff research scientist from Institute, and I'm going to kind of take us through the first part of the discussion today, right? Obviously, we know where we're heading, this kind of flexible antibody multimodal model, but I'm going to kick us off with maybe a bit of an introduction just for what we were actually trying to achieve with these foundational models and maybe why we ended up picking Bayesian Flow Networks, which may be a bit non-standard, right? I wouldn't be surprised if there's people here who are watching who are maybe not as familiar with these as they are other paradigms. So a bit of an introduction to how they work, why they're fit for our purposes. And then I'll accelerate into biology and hand over to Bora to take you into the world of antibodies. But with that said, I'm actually going to start from a very, very high level, right, with a couple of hopefully non-controversial statements that really fundamentally what we're trying to do with AI, especially kind of in this scientific domain, is learn useful functions from our data, whether it's classification tasks, regression, or more exotic paradigms. And all of these are some form of generative task, right? You're generating some set of variables based on some other conditioning set of variables. But the conventional approach, if that's the right term, is that you would pick the task that you're trying to solve, you would curate yourself a data set, train a model, validate it, and use it in the pipeline. So pick an example, something like AlphaFold, right? You want big protein structures, you go to the PDB, you fish out the training examples, train your model, and then that exists as a useful tool for scientists, but it's not, in the application sense, it's not the be-all and end-all of what we'd want to do, right? We want to have an ability to kind of go back to our models, interrogate aspects of them, maybe have it not just learn to predict a structure, but learn the relationships between sequence and structure, or between sequence and structure and other metadata modalities that you might be interested in. And so the vision, and this of course is not uniquely our vision, it's the vision of many, many people, is to train these kind of broad foundational models on heterogeneous scientific data, where quite agnostic to what you want to do with them, you'll just pour all of your data in, you have your model learn the joint distribution across all of that data, and then in some way at inference time, you can access the conditional distributions to define a task, right? So you can use the data modalities that you defined as some sort of syntax for defining useful tasks at inference. And so to make this a little more concrete, here is my very limited example, example of what this might look like for a proteome, right? For proteins. So you have classic modalities, sequence and structure, but you also have a lot of metadata associated with these, whether it's functional annotations like Go terms, EC terms, enzyme commission terms, the organisms, post-translational modifications, or even binders, assays, experimental results from a lab. And the statement to bear in mind for this talk is that out there does exist some joint distribution over all of these variables, right? It might be super complex, it might be completely intractable, but it does exist. And what we want is to find a model that can learn that joint distribution and that we can then access conditional subsets of it at inference time. So taking the protein folding example, I would be able to say, well, here's a sequence, generate me a structure and just marginalize out, forget the rest of the data modalities, they don't matter for this task, right? Or I could do something different, I could give a sequence and structure, I could give myself, and annotate with some Go terms for some functional annotation, and obviously combinatorially many options of what you could do in here. But this is obviously a nice aspiration to have, but in practice, you have to ask yourself, how do you build a model that can start to approach it? And especially for kind of scientific data, it's really heterogeneous. There's often sparsity in the data, and there's clearly going to be different data modalities: continuous data, discrete data, discretized data, and in an incomplete taxonomy of different generative models, there isn't a single framework that really naturally handles all of these things together, right? So, diffusion models, classic adding Gaussian noise to some continuous variables and having a model that's trained to denoise it, right? Work brilliantly for continuous data, state of the art for images, allows you to do a lot of these inference time tricks that I'm trying to get towards, right? Where you can have an unconditional image generation model, but you can turn it into something conditional So you can have it inpaint it with classifier guidance or classifier free guidance. So you can have this kind of zero-shot conditional generation. For biology, you see these obviously for continuous data domains, so protein folding, structure prediction, things of this nature. But they don't naturally handle discrete data, right? It's not as obvious to say, what does it mean to kind of smoothly noise and denoise a discrete variable? The kind of king of generative models in that task is in that domain is GPT models, right? Generate everything left to right, great for de novo generation, have something completely new, but maybe less clear how to apply them for kind of arbitrary conditional generation because they have some generative order. So you can't just mask out certain regions that come before one variable, ask you to predict it. If you wanted something more towards that flavor, you would look toward mass language modeling, the BERT models, kind of ESM models in biology, kind of classic example here. And these are often used for representation learning, really strong for discrete data, but not as clear how to, out the box, do de novo generation, make something completely new, because they're often trained to predict part of the input from the rest. But what all of these models have in common is that they follow the same basic paradigm, right? They kind of noise and predict. You take your ground truth data, you noise it or you perturb it in some way, and then you train an ML model to try and undo it in a neural network. And from that, you can extract some sort of generative process. And so we followed in these footsteps, but we said, well, actually, none of these are really giving us exactly what we want. And so we ended up picking Bayesian Flow Networks. Now, Bayesian Flow Networks first proposed back in 2023 by Alex Graves, and we're kind of lucky enough that he's worked with us on everything we're going to talk about in this as well. And they were designed explicitly to try and unify this. I don't know if it's unify this gap or bridge this gap or something in the middle, but to be a natural framework for kind of unified multimodal modeling. There is kind of 80 pages of maths that goes into this that we're definitely not going to get into in this talk. I can leave that to Alex's original paper if you want it. But to give kind of the cliff notes and the kind of the motivation for it, rather than directly modeling the data, which is kind of what all the things we discussed so far are doing, what a BFN is modeling is a distribution over that data. And the reason that's really powerful is because whilst the data itself might have different modalities, continuous or discrete, the distributions will always be parameterized by continuous variables. So for continuous data, it might be means and variances and Gaussian distributions that you fit. For discrete data, it might be the probabilities or the logics, depending on the space in which you work. And so you can start to see how we can have this smooth noising, denoising process, almost a diffusion-like process on the parameters of your distribution rather than on the data itself. The point here is then you can naturally treat all these different modalities as kind of so-called first-class citizens, so continuous, discrete, discretized. And that makes it, we think, we still believe, kind of ideal for this use case that we've laid out. To give some intuition for how this works behind the scenes, the original mathematics was derived from this noisy communication sender-receiver protocol. Right, this is what you'll find in the original paper. So for people who are familiar with this, you often get kind of this notion of an Alice and a Bob who are talking to each other. And the idea here is that Alice has your ground truth data. I've picked binarized MNIST for this, just as visually easy. And Bob has no idea at the start what that data is, but he's going to try and get information sent to him by Alice. In our case, Bob's a good Bayesian. So every variable is a pixel, and he has a uniform prior of what they might be, doesn't have any assumptions. And Alice is going to send some noisy example to him, and he can do a closed form Bayesian inference, right? Nothing AI here. To do a per variable update and update his belief of what Alice might be having. And you can repeat this process, sending increasingly informative samples, and eventually end up with Bob having a really strong idea of what the ground truth is. The trick is then how do you turn this into something generative, right? Where's the neural network come in? And the high level here is that just like in the diffusion case, what Bob's going to try and do is always predict what the ground truth is based on what Alice has sent him so far. There is a kind of asterisk here, or so certainly it was in my mind, I started thinking about it, which is if Bob is doing kind of Bayesian optimal inference over these variables, what is there left for him to predict, right? Why can he actually do any better than his prior distribution at a certain point in time? And the rationale there is that this is a per variable Bayesian inference, right? So every variable is just getting updated based on what information is getting sent. But what Bob can do is look across all the variables and learn correlations between them, and therefore try and improve the prediction for one variable based on what's been sent to the other variables. So classic example of this in images, the colors of different pixels will tend to be correlated if they're spatially grouped, right? So if all the pixels around you are white but your Bayesian inference is taking you to a point where you think you might be black, you're probably going to want to update that belief after having learned what everything around you thinks their color is. So this is the intuition, and we can train this neural network by what we do, KL divergence, you want to call a sender and receiver distribution. This really is just the probabilistic equivalent of an MSE loss that you'd get in a diffusion model. So because we're modeling distributions as a KL divergence, but it's just saying, how well is Bob modeling what the ground truth is under this noising process. And you can train this at any different step in time, n different steps for your generative process, and then have a model that learns this so-called Bayesian flow. To be clear, whilst I've put this out as like discrete time step process, obviously you can find closed form approximations for these things. In fact, not approximations, closed forms for these. And so you can have a continuous time equivalent of this loss, and it's trained a lot like a diffusion model where you sample a step in time, sample your noise, train the difference between the sender and the receiver. So that's the crash course in BFNs. As I say, there's a lot more math that goes into this in the paper. The thing to bear in mind is that we're modeling distributions over the data rather than data itself, and that this is helping us with our multimodal modeling by treating everything in the same framework, right? That's the takeaway if the rest was just pretty pictures. With that said, before I get to the antibodies and Bora, I'm just going to talk to you a bit about how we validated that this was a sensible approach, right? And that is really what this first paper is about. It's about saying, well, we have this idea that Bayesian Flow Networks can be really useful, but let's check if that's true. And so we specifically picked protein sequence modeling because in the continuous case, if you want this kind of flexible model with conditional generation inference time, you will train a diffusion model, and BFNs are very close to diffusion models on continuous data. It's the discrete case that is kind of more interesting, because for different generation tasks, there are different paradigms, different generative frameworks that kind of lead the way. If you want to do de novo generation of a protein, you might use a kind of autoregressive model like ProtGPT from earlier for us. You might also try and use a discrete diffusion one from Microsoft like EvoDiff. But if you want to do conditional generation, kind of inpainting of one part of a sequence from another, you would use your ESM models, right? It's something that's been trained explicitly with that BERT loss. And so, long story short, we took these different tasks, we trained our BFN on them, and we wanted to evaluate, well, do we learn the underlying distribution really well? Do we get good natural looking proteins? And the answer is yes. I'll quickly on the next slide unpack how we validate that and what that means, but the answer is yes. And can we do this zero-shot conditional generation? Can we access arbitrary distributions from within there for more task-specific inference? And again, the answer is yes. And this paper is out online now if you want to go and dig into it in details, because I'm only going to very quickly skim through it here. Just to put some meat on the bones, this is maybe like the quick model card, so to speak. We curated our own data set, 72 million protein sequences, 20 billion amino acids. An asterisk here is, yes, we did ablate the differences between the data sets of us and the baselines. And the differences that we see are to do with the algorithm, not just to do with better data curation or anything like that. We trained what's probably quite a small model by today's standards, right? Only 650 million parameters, but a big batch size, 8,000, and trained for a week or two on a bunch of TPUs. I won't go into this in more detail, but it's just to make a bit more concrete what this actual model looked like under the covers. The first test, of course, finally, after talking about this kind of new generative paradigm, is to say, well, how well does it generate things? Do they look like the underlying training data? So in this case, does the proteins that ProtBFN generate look like natural proteins? And it's actually kind of an interesting question, because there's not a single metric for a good protein generation, right? You want them to be natural, you want them to look reasonable, you still want them to be diverse, right? And you want them to be novel. You don't want to memorize your training data. And so that's kind of what we're trying to show here. We took our model, ProtBFN, we took a discrete diffusion model, EvoDiff from Microsoft, and an autoregressive model, ProtGPT2, generate 10,000 sequences with each, and then we analyze them in a few different ways. So for naturalness, we are comparing to, in this case, UNIRF50, so natural repertoire protein sequences, and saying how often do we get a hit in that database, so 50% sequence similarity. And what we're seeing is that our model is generating hits at a much higher rate, right? We're generating more proteins that look kind of natural, that would have been clustered into an existing cluster in UNIRF50. So that's a good sign. But obviously, you could just generate the same protein, the same real protein over and over again and get 100% on this metric. So you want to look at diversity as well. For diversity, we derived a new metric that we called a coverage score. But really what this is saying is, if I was sampling from the true underlying distribution, how many different clusters in UNIRF50 would I expect to hit after generating n being 10,000 here samples? At a very high level, 0 would mean that you just hit the same cluster every time. And 1 would mean that you're kind of perfectly getting the diversity, you're spanning the diversity, you're getting as much breadth as you would expect. We see that we're not at 1, right? So we still have some preferences in our model towards specific clusters, but far less so than other models, right? We still have much more diversity in what we're generating. And so that's, again, a really good indicator. And then the last test, of course, is both these metrics would be perfect if we just replicated the training data and just memorized it and generated the samples. So we have to also look at the novelty, right? Just make sure there's nothing in our training data that looks very much, that identically matches our generations. And the answer is we are novel, right? We're highly novel. Almost just under half of our generations have, very, very low sequence identity. So really no homologs in our training data, despite still kind of being quite natural and diverse. And so that's great, right? Again, the takeaway here is that a BFN does seem to be doing a strikingly good job at being a generative model for these for protein sequences, right? For these discrete variables. It's also worthwhile because we're interested in the biology as well as the AI to analyze these generations not just in the sequence space but in the structural space. Because, as I'm sure many of you know, kind of the sequence, broadly speaking, the sequence is determining the structure of your protein, the structure is determining the function. And so what we want to do is check that we're actually getting, good structural generations from our models. Even though it's only generating the sequences, right? The structure is a function of the sequence, but there may be regions in structural space that are very close together that are very far apart in the sequence. And so to get well-generated structures, you need this kind of global coherence over the entire protein sequence that you've generated. And so to do this, we take a different model, ESM fold in this case. It could have been AlphaFold. ESM fold was just a bit quicker. Predict structures for all of our generations, and then do a very similar analysis where we match up to the known structural database, in this case, CAF-S40. And again, we get much higher rates of getting structural motifs that look natural, right? Things that have a hit in this database, hit being a TM1, TM2 above 0.5 for people who kind of want to get into this. And when we do have a hit, those hits tend to be higher quality as well. It just means that they have greater structural similarity because a hit is just a threshold, right? It's not a measure. Once you're above that threshold, it doesn't tell you how good you are. Again, takeaway here, this looks really good. We're generating unconditionally very well from the model. And we can really get into this and start to look at biology a bit and talk about how many single and multi-domain proteins that we're generating, right? We're generating complex proteins with up to 4 different domains within them. And if you classify these, in this case using CAF, so annotation of protein structure, we see that we're spanning the diversity of the different classes within CAF and the tree of life, right? So different secondary structure elements, alpha helices, beta sheets, irregular domains, small and large domains, transmembrane and enzymes, spanning different elements of the tree of life, human, bacterial, and things like this. This is really just a pretty picture section, but the point here, again, is that we're getting really effective unconditional generation of the protein sequence. Now, the next part of this is to look at that conditional generation, right? This tells you we've learned the underlying distribution really well, but it doesn't say whether we can then do something useful with it. So we do have a whole section in the paper around kind of doing this inpainting task on antibodies, on protein sequences, and showing how well that works in different cases. I'm going to completely skip this because this is basically the ABBFN1 model, and Bora is about to talk about ABBFN2, which is frankly much more advanced and much more impressive. So you can take my word for it. The kind of kicking of the tires test did work very nicely for the sequence-only models. But rather than going into those details, I think it's better just to go straight into BFN2, where we get back to that original picture of multimodality, and we see how these things work in a more applied setting, rather than in the setting I've been talking about, which is really more just testing that it works as well as we hoped it would. So with that, I'll pass to Bora, who's definitely the expert in this domain. And yeah, take it from here.
Cool, perfect. Thanks, Tom. Yeah, hi everyone. I'm Bora. I'm a research scientist at InstaDeep, and my background is more in immunoinformatics, where I kind of looked at structural flexibility of antibodies. And so I kind of started working on ABFN2 since I joined InstaDeep. And what we really wanted to do with this model is kind of have a good multimodal model that allows you to tackle many, many tasks, right? So antibody design is a classic multi-objective optimization problem, right? You have the key thing, obviously, is affinity, but oftentimes a discovery campaign will start with a kind of precursor antibody that actually has relatively good affinity. And then you go through these repeated loops, these design loops of trying to optimize the antibody for developability. So this is in the therapeutic design context, right? So you might need an antibody that has good levels of hydrophobicity. You don't want it to crash out of solution once it's bottled up. It needs to express really relatively well, so it needs to be stable. It also needs to kind of not elicit an immunogenic response once it's administered, and so on and so forth. It kind of what the tasks are going to be really depends on the exact process, right? If I'm starting my discovery campaign with a human library, then I don't need to worry about humanization. But in that case, I will have to worry about biophysical optimization and so on and so forth. So it really, really depends. And that's really why we think this sort of model shines. This task agnostic model is really good for these sorts of problems. So in this case, that task agnostic nature of it comes through the fact that we can just condition on any sort of data that we want, that we're interested in, any attribute, and then we can just generate based on that. And that really means that kind of we can go beyond the general convention of you have n models for n tasks, and you apply model 1 first, and then model 2, and then model 3. The risk with that is essentially that you might run the risk of, because the models are unaware of what the other ones have done previously, you might run the risk of undoing a mutation that you've previously introduced that is actually critical for some sort of optimization. Right, so concretely what that looks like is essentially we've taken the antibody and we've broken it down into these 45 data modes, we call them. So these span both the sequence of the antibody. Mind you, we're working with paired sequences here. So that we break down into the CDR loops. So those are the kind of colored bits in that diagram there. They're the really important bits for binding. And then also the framework regions. So those are the grayed out regions. And then beyond that, we also look at stuff like biophysical attributes that are therapeutically relevant. So hydrophobicity, positive, negative charged patches, and then charge imbalance across the heavy and the light chain. So the heavy chain is that the left region, so green, blue CDRs, and then the darker kind of framework regions. And then the light chain is the region on the right. So the kind of ochre tones and the lighter gray. We also look at genetic attributes. We know that certain genetic lineages of antibodies are better suited for use as a therapeutic. We also look at the locus of the light chain. So light chains come from two loci on the chromosome. So you have the kappa and the lambda light chains. Species as well. Lots of antibodies will be generated from human, or rather sequences will be known from human repertoires, but there's also a lot of data from mouse and rat repertoires out there. And then what we're also interested in is CDR length. So generally a shorter CDR length is preferable for therapeutics just because it makes the thing more stable. So once we have all of these kind of 45 data modes, we essentially have generated a really rich syntax or grammar that allows us to interact with the model, right? So if we think about it, these kind of sentences that I've provided here, given the CDRH3 sequence and this light chain V gene, I would like a human developable library of sequences. These are all tasks that kind of we encounter quite often in the design process, and they can all be expressed using a combination of these data modes that I've described. So if I give a kind of properly concrete example here, say you start off with this antibody here on the left. So you're happy with most of it, but that CDRH3 loop, so that green loop, is far too long. You want to have a shorter one of that. So what you can do is just condition on the data that you already have and that you want to retain, right? So the rest of the sequences, you can see that the CDRH3 is gone now. And also, because we explicitly model the CDRH3 length as a data mode, we can also condition on that. When we query the model, it then returns a new antibody sequence. These are actually generations from the model. I've just folded them up. Returns a new antibody sequence with a new CDRH3, and everything else, all of the other conditioning information is respected. So again, you're generating a distribution of CDRH3 sequences conditioned on the information that you already have, in this case, the sequence, the remaining sequence, and the CDRH3 length. So then the task might change, right? So maybe the next step is actually, okay, I have this heavy chain now, this is great, but I notice that the hydrophobicity of this antibody overall is off. And I also don't like the LV gene. So the V gene is kind of the major genetic modality that contributes to each sequence of the heavy and the light chain. So maybe I have a specific LV gene in mind, and I also want to optimize the hydrophobicity to be in line with known therapeutics. I can do that. I can now condition on the heavy chain sequence, the LV gene, and the hydrophobicity, and again, I get a new light chain out. So the heavy chain has been respected again. So that really kind of is to showcase that flexibility. But before you can actually tackle these interesting tasks, you need to make sure that your unconditional generation works, right? If the unconditional generation falls over, then there's no point in actually conditionally sampling the model. And we've done this in a number of different ways. It's always difficult to kind of assess how well a generative model works because it's new data, right? You're not just comparing to kind of a holdout set or to hold out labels. But in this case, we started just by generating 10,000 sequences. And then we looked at the CDR length distributions first. So the gray OAS data here is holdout paired natural sequences from the observed antibody space. And then the blue are generated samples. And we find essentially excellent agreement here. So on the CDR length front, we're all fine. So we took it kind of to the next level. We folded these sequences up and then also folded the holdout sequences up and structurally compared the CDR loops. Now, anyone who's worked with antibodies will be familiar with these plots here. So the so-called minor loops, that's every CDR apart from CDRH3, presents itself in these so-called canonical forms. We find specific shapes in the PDB, and these are repeated. For CDRH3, because it has much greater sequence diversity, it actually is very, very unique in structure as well. With, I think, 30% of CDRH3 loops in the PDB are actually novel structures. Whereas for the other loops, we expect to find these kind of known clusters. And we recapitulate these as well, right? So especially for L1, for example, it's very visible there that we kind of get the same peaks in this distribution across both the OAS, so the holdout data, and the generated samples. We also take this a step further. So when we actually look at other data modes that we model, so we, alongside kind of the germline genes, we also, for example, model the germline identity. So how close is this antibody to the progenitor sequence, to the naive version that it was kind of matured from? And this is a continuous data mode, for instance. And here again, we see that we kind of match that natural distribution really nicely. So that's on the top right. On the bottom, we have biophysical attributes that we're interested in, right? So going from left to right, we have hydrophobicity, positive charge density, negative charge density, and then charge imbalance across the two halves of the antibody. And again, that distribution is also matched very well. I should note here that these are actually predictions. Sorry, can we go back one, Tom? Thank you. I'll note that these are predictions that are made by the model, right? So they're not ground truth values that we've accessed from the sequence. But when we actually compare these predictions to, the predictions from the model to the kind of ground truth that we can generate with the sequence that is co-generated, then we actually find excellent agreement here as well. So these are correlated with the sequence, which is exactly what we want to see. And then lastly, in the top left there, I have kind of the different gene pairing. So different types of genes will preferentially pair with other genes. And we also find that we kind of recapitulate those pairings really well, which is really nice to see. So kind of having shown that the unconditional generation works really well, we can move on to conditional generation. Now, the easiest thing there is just show that sequence labeling works really well, right? So condition on the sequence itself for a holdout set, and then generate all of the other metadata or the other data modalities and compare those to ground truth values. Across 23 tasks that we've tackled here, we find that ABFN2 is state of the art on each one. We're not kind of breaking any new ground here, right? So the improvements are when they're modest, but that's also not necessarily what we're after, right? So for instance, we're comparing gene identities to IG BLAST here, which is a much, much, much simpler algorithm, right? But the key thing here is that if we can learn all of these different attributes together, then we can tackle the inverse task, right? So then you can actually look at multi-objective optimization. But first, you need to actually make sure that sequence to attribute works really well. There's a couple of things to note here, which is the prediction of the yeah, there we go. The HJ gene, for instance, goes up by a fair bit. So that's on the bottom there, towards the bottom in the classification tasks. We get an improvement of 0.1 on the F1 score. And that actually is, we think, because the model has access to all of the data, right? So when, for example, BLAST looks for an alignment, it just looks at the sequence that actually aligns with the database search. Whereas here, the model can actually make use of the rest of the sequence as well. And because, for instance, certain V genes will preferentially pair up with certain J genes, then that knowledge can be incorporated in the prediction of that J gene. So there's kind of nice emergent properties of these predictions that we were very happy to see. Going beyond this, these kind of annotations we find are also functionally relevant, right? So here we're looking at a set of roughly 200 antibodies that have gone into the clinic. And so we actually have functional readout for these. And the readout here is in the form of anti-drug antibody responses. So it's the percentage of patients who were administered these antibodies and they developed an immunogenic response. So we want to be low here. These are publicly available sequences that were collated at some point. And we find, for instance, that when the model thinks a sequence is very, very human, so when it confidently classifies a sequence as human, those are the sequences that have lower ADA values. So on the left, there's kind of a correlation plot. And then on the right, we can really see when we bin these things, a much clearer picture emerges. So the really confidently human sequences are the ones that are low risk. And then all of the high risk sequences actually fall into the confidently not human bin, so to speak, under the model. But the kind of functional relevance of the learned distribution also goes beyond this. So we can look at things like redesigning VHVL interfaces. This is really important if you want to stabilize an antibody, or if you have a heavy chain and you want to repair it with a different light chain. So in this case, what we did was we masked out the interface residues. We just condition on sequence here, but partial sequence. We mask out the interface residues and we just regenerate those. And we find that the model actually regenerates them really nicely. So the interesting point here is that amino acid recovery is only at 75%, right? So we're not predicting the exact same interface each time as the input antibody. But in terms of the energetics between those two halves, the distribution is virtually the same as the holdout distribution, right? So the background distribution is still there. So functionally, actually, these kind of sequence recovery tasks are very meaningful. We also have a negative control there that's just VHH is paired with a light chain. And in this case, lower is better. So that's kind of annotating the sequence in kind of these single objective tasks, right? But the point of the model really is that you should be able to tackle multi-objective optimization. And here we have two flavors. So that's afforded to us by the flexibility of BFNs, right? You can either look at kind of conditional generation or complete de novo generation. We'll tackle conditional generation first, so inpainting. And in this case, we start off with a set of 91 antibodies, again from the holdout set, that have at least one biophysical liability. So either their hydrophobicity values are off compared to known therapeutic antibodies, or their charge densities are off, or the charges are imbalanced across the heavy and the light chain. And they're also from non-human sources. So they're mostly mouse, sometimes rat antibodies. And what we want to do here is get into, so for panels A to D, we want to get into that light blue region. That's the region that known therapeutics occupy on these developability metrics. And then on panel B, we just want to go from 0 to 1. So the precursor antibodies, the starting points are all in the gray histograms, and you can see that kind of the distributions are not where we want them to be. We used BFN2 to redesign these, and when we're redesigning the sequences, we're actually using a relatively kind of complicated sampling algorithm. So we found that kind of just conditioning on the CDRs we usually want to keep, so we condition on those explicitly. Just conditioning on those and then regenerating a whole new framework doesn't really work. And it's also not usually what we're after. What we're after is kind of point mutations. We want to introduce as few changes as possible in order to kind of get the objective, but not massively change the precursor antibody. So what we do is we mask, we condition on the CDRs at all times, and then we also partially condition on the entire framework. So the model sees the whole sequence for the first n steps of sampling, and then we make it invisible. So that way, if the model really thinks this position here, if I change this, that's going to make a massive impact, then it can absolutely change that, but it's still incentivized to keep the original sequence as much as possible. And we then use the output from each iteration to feed into the next iteration. So that's a classic kind of recycling scheme there. And we find, as expected, the more iterations you need to actually optimize these sequences, the more total mutations you get, but it kind of plateaus out at around 60. And that's for the really difficult cases. In most cases, we actually get there in a single iteration, so we don't even need the recycling scheme. And we see that in almost all cases, we manage to optimize all objectives, right? So we call it a success if we can then look at the sequence and then calculate these values, so the ground truths, and that are actually what we want to see. And in 80% of the cases, we can do this. But there are some antibodies for which this doesn't want to work. So if you look at panel C, for instance, the PNC, so the negative charge density is often unchanged in the final designs. So we had a look at why this might be the case. What are these antibodies that are failing on this? And we actually found that those are antibodies where the CDR loops themselves are already highly negatively charged, much more so than all other antibodies. And so we're telling the model in this case, I don't want you to change the CDR loops, which are highly negative, but I also want you to make the overall antibody much less negatively charged. So there's kind of this paradoxical prompting going on in these so-called intractable sequences. And so the model eventually is going to fall over, right? This is really to kind of hammer home the point of when you have these really complex models where you can really be very kind of creative with how you sample them, a certain level of domain expertise is really required to kind of engineer the prompt well. You need to be able to look at what you're inputting into the model and whether that makes sense within the domain. So when we actually take out these kind of intractable sequences, the success rate shoots up to 90%. So that was redesign, but we can also look at de novo generation. So say, for example, you had a set of kind of conditions that you wanted to satisfy, but you don't have a precursor sequence yet. Maybe you want to generate an initial library that you then want to fish from with some antigen. So in this case, our test case is, or our case study is VRC01 type antibodies. VRC01 is an exceedingly rare type of antibody. It was first discovered in 2001. And what's special about this antibody is that most people, due to their genetics, are unable to produce these antibodies naturally. But when they do, HIV patients that are able to produce this class of antibody can control infection much better than those patients who can't produce them. The reason why they're so rare is because they kind of present a combination of attributes that we never really find together. They're already rare by themselves, but in combination, they're exceedingly rare. So there's a specific light chain locus that these always originate from. They're always kappa light chains. The CDRL3 length is really, really short. It's 5 residues, which is really rare in natural antibodies regardless. There's a specific HV gene lineage that these have to come from. It's IGHV12. And there's also a set of mutations on the framework that kind of we find in these antibodies and one on the CDRH3. So those are the ones in purple. So positions 55, 66, 80, and then on the CDRH3, it's 113. The reason these mutations are there is actually when you look at the structure of the original VRC01 in complex with the antigen is that they make interactions with the antigen via those residues. So when we look at the entire training data of the model, so all of OAS paired sequences, that's 2 million, we actually only find that there's 21 sequences that have all of these attributes together. I should note here that 10,000 of these sequences actually come from HIV patient repertoires. So that really kind of gets home the point that these sequences really are very rare. With there being only 21 antibodies, so normally the way kind of we would achieve this distributional shift, right, in an autoregressive model is we would fine tune on what we want to generate. But when we have 21 examples, it's very difficult to do that. With ABFN2, what we can do is essentially just condition on the things that we're interested in. So we can condition on all of the things that I've listed below. And we find that the distribution has shifted really nicely. So we generated 2,500 examples here. And we found that 1,700 of those satisfy all of the criteria that we asked for. And they're also all unique. So none of the sequences are repeats of each other. And they're not repeats of the sequences that we find in the training data. So they're all actually distinct sequences from that training data as well. So that kind of is a different flavor of the model, right? So that is kind of conditional de novo generation. So if I kind of pull that all together, what we've done with the model is we've trained on sequences and the associated metadata that might be of interest. And that again provides this rich language that we can speak with the model with. That comes also with the kind of, it's a double-edged sword in that that comes with the need for correct prompt or task engineering, right? If you ask things of the model that are paradoxical, it's always going to have trouble. But that's because the domain just doesn't work like that. Antibodies don't work in a certain way, so you can't request antibodies that are like that. The architectural flexibility of BFN, so the fact that they can deal with all types of different data and also that you can tackle both de novo generation and conditional generation, allows the model to essentially kind of fit into multiple different tasks, right? So it's very task adaptive. And because we can combine these tasks into one, that also means that kind of these traditional pipelines of apply 1, 2, 3, 4, 5 models, one after another, get compressed into a single step. And then lastly, this kind of, as we call it, the condition anywhere, generate anywhere paradigm means that the model actually admits a wide variety of tasks that you might see in the kind of antibody design space on a day-to-day basis. So that also means that if the task changes at any point, the model doesn't have to